right, good evening, everybody. Thank you all so much for coming out tonight. Before we begin, I just have one small housekeeping request. If you need to leave during the presentation, please use the doors in the back of the auditorium and just head upstairs, so we don't make a disruption on this side. But for those of you who don't know me, my name is Jeff Brown. I'm the director of the honors program, and I'm so excited that you are all out here for our first honors series event of the 24-25 academic year. I know that the first round of exams have hopefully passed for everybody, and I really hope that this is a chance for us to reconnect and kind of engage with some interesting ideas and hopefully get some inspiration from our speaker, Dr. Jan Spacek. One of the main pillars of our honors program here at Embry Riddle has always been interdisciplinary thinking. And I've had the opportunity to spend a lot of time with Dr. Spacek today and a lot of students, and it's very safe to say that he is an exemplar of interdisciplinary thought and trying to figure out how to do amazing things like molecular biology on another planet. So if you plan to do that, you're going to need to know a little bit about rockets, you're going to need to know a little bit about instrumentation, but most importantly, I think you need to be able to also convince somebody that it's worth doing. And he's been very, very intriguing to hear his thoughts on this. So before I formally introduce our speaker, though, I wanted to take just a moment and provide some background on the honors series events. We've been doing this now for over 22 years. And during this time, we've hosted over 100 authors, public intellectuals, performers who really embody the ethos and the spirit of the honors program. So all of that's very important, but none of it would really be worth anything if it were not for you all being present here tonight. So thank you for being an active part of our community. To introduce our speaker, Dr. Jan Spacek is a senior research scientist at the Foundation for Applied Molecular Evolution. His work spans molecular biology, analytical chemistry, organic chemistry, and instrument development, with a focus on the search for life on Mars and organic chemistry in the clouds of Venus. Dr. Spacek is the founder and leader of the ALFA Mars group and the inventor and lead designer of the Agnostic Life Finder. He holds a doctorate degree in genomics and proteomics, along with bachelor's and master's degrees in molecular biology and genetics. Dr. Spacek has proposed a hypothesis regarding the organic carbon cycle in Venus's atmosphere, which will be tested during the first private interplanetary mission in 2025. He also works on developing life detection devices, including tools to detect the coronavirus. His extensive research experience includes work in analytical electrochemistry of DNA at the Czech Academy of Sciences and the Central European Institute of Technology. Please join me in welcoming Dr. Jan Spacek.
Great, mic works. Thank you very much for coming. Thank you for the very nice introduction. I would like to thank Amy Williams and Jeff Brown to inviting me, and for Courtney Pasternak to organizing this. It's an honor to be on the honor series. And thank you all for coming. I hope I'm going to entertain your thoughts tonight. So, okay, I'll use this. I will, in my talk, discuss our collective journey. I will make a brief overview. It works. I will make a brief overview of what humans accomplished in exploration of Mars and search for life on Mars. And then I'll shift to my personal experience, what I'm trying to do. So I would not be looking for life on Mars if I did not believe that there's a chance that we can find it there. So why do we believe that life can be on Mars? Simply, through analogy, everywhere we look on Earth, we can find life. We can find it in a dry permafrost, multiplying well below the freezing point without excess of oxygen. and living in perchlorates, which were found on Mars and thought to be maybe detrimental to life. We can find ancient life in all these glaciers we found on Earth. Basically, the life was deposited on the surface of those glaciers 8 million years ago, and then was buried with overlayers, and it's still surviving without communication with surface. That's analogical to many sites on Mars. We can find life in deep subsurface, basically everywhere you dig down, as long as you have access to some water through percolating through channels, we can find life. And in very dry regions like Atacama Desert or dry Arctic soils, we can find life, and we can also find it. The record so far for observed replications of bacterial cells is at minus 18 degrees Celsius. You would think that once it freezes, it stops, but they manage to survive even and replicate in that space. And recent discoveries, we found that life in Antarctic dry valleys actually uses trace gases like hydrogen, which is in parts per million in atmosphere, and they oxidize it with oxygen, making water. They use the water for their metabolism, but also take the energy from basically burning the hydrogen to produce energy to capture carbon dioxide and to live off that. And so the more we study, the more we are quite convinced that it would not be a surprise to find life on Mars. Mars used to be much more clement. 3 billion years ago, there were rivers, there were lakes and perhaps ocean in the northern hemisphere. And then the Mars went through a slow transition to dry, cold environment we see today. But the story doesn't end there, and I'll get there future. Surprisingly, except for Viking experiments in 1976, well, let me say this. So except for a short period after the Viking experiments in 1976, people have made the assumption that there is life on Mars. I'll discuss the Viking a little bit later. So, which is right now the brief history of in situ exploration of Mars. Here's the timeline of the probes we sent to Mars. Notice that at the beginning it's very red. That's the Soviet Union sending the probes, and then NASA realized that, we are losing this part of the space race. We should do something about that as well. So they got the Viking in, and that's too confusing. They got the Viking in 1975, that was launch date, and 1976 when the Viking was on Mars sending out the results. And here are the landed probes on Mars. It misses one because simply it didn't fit into the lineup. That's the only reason, which is Chinese probe. But at the time, in 1976 and 1975, NASA claimed that the search for life on Mars might be the greatest experience in the history of science, which is very interesting. I still believe that. So here are the future plans of year 2026 and on. You might notice that Mars sample return, that's the MSR, it's a still unknown date. It's been postponed, it's been, maybe it's going to be reinvented, who knows. But the Chinese sample return, it will be happening in 2028, and Chinese Space Agency is very good in sticking to the timeline. So it's quite possible that the sample return will look like this. There's a joke I made in April 1st, 2022. Can save NASA quite a lot of money if they wait just a little bit longer, because there will be no point in actually doing the sample return because we will have humans on Mars already. So, but I want to discuss one thing, which is the Viking gap. You can see a lot of missions until Viking and then nothing for almost 20 years. So the reason why Soviets didn't go is because the socialistic experiment was falling apart. The reason why NASA didn't go, I think that's because the Viking experiments and what they found. So what did we saw with Viking, which made us not send any more experiments? There were 3 biological experiments on the mission and 1 gas chromatography mass spectrometer, which was not originally meant to be a live detection experiment, but later was very significant. So the first experiment was gas exchange, when you took a nutrient broth, then you fed it to soil on the Mars, and observe the exchange of gases, right? If you intake something, you breathe out carbon dioxide, or you can produce other gases. They observe release of carbon dioxide and oxygen, which was surprising, because then you observe that when you have actually life in the sample. What was surprising, that after heating the sample to 160 degrees Celsius, the gas release was slowed. It was still present, but it was hard to interpret at the time, but at the first, they interpreted it as positive result, but later they revised it as a possibility explained by biologically with superoxide. Surprisingly, we still have not found these superoxide they first proposed, so this result is still inconclusive, and we still don't have a good explanation either way. Then pyrotic release, that was experiment which basically was testing photosynthesis, where you provide to plants light and carbon dioxide. In this case, it was labeled carbon dioxide with C14. You observe fixation of that carbon into the plant, and then when you burn it, that's the pyrotic release, you can observe the carbon dioxide getting into gas form, which you can detect with your detector. Again, initial results were interpreted as positive because we observed the radioactive gas release. Later, it was debated and then either disputed or inconclusive. And the last of the 3 biological experiments was label release, where you put, again, radioactively labeled nutrients. You expect bacteria to consume them, use them in their bodies, and then expel, for example, carbon dioxide or methane. The controls in these experiments were very interesting. So this experiment was positive for when they first introduced the organics. The bacteria actually, or the bacteria, the soil samples released the gases which were radioactive, which we detected, but the controls, even when you heated the sample, and remember, these were done in duplicates in 2 rovers on different sites, and all of these provided these results. So if you heat the sample to 150 degrees, the assumption is that you kill all life, and then you would not observe the release of the radioactive gases. That's exactly what they observed. Then to make sure that it's actually not some chemistry, they heated up the sample just to 50 degrees. The assumption being that no chemistry would be deactivated at 50 degrees Celsius. But what they observed is that the release of gas was diminished by about 70%. And then last interesting control, they just store the sample at 10 degrees Celsius for about 2 months. And then after 2 months, they did not observe any release of radioactive gas. So this is super interesting. There is no explanation of this behavior without using biology today. And yeah, something to think about. But then we had a GCMS, mass spectrometry, which did not observe any organics in the soil. So the logic is that all life, as we know it, needs organic material to build the bodies, to use for metabolism. And if you don't see any organics in the soil, that means that you cannot have any life in that soil. So based on this result, the official overall result of the Viking experiment was concluded as a negative, that Viking did not find life. So, but since then, since 1976, we know that there's organics in the soil. So the reason why the Viking did not detect them, there are several reasons. One is that on those sites were probably perchlorates. So if you heat up the organics with perchlorates, you basically burn them. But also the GCMS was not sensitive enough to detect cells at a high enough sensitivity. So the LOD, which is limit of detection of GCMS, would be about 1 million cells per cubic centimeter, which is quite a lot. You have that in quite fertile soils on Earth. But if you study some more arid soils, which are analogous to what we see on Mars, you observe like 100 cells per cubic centimeter. So the label release had a limit of detection of about 10 cells per milliliter, so that could be the discrepancy. And second thing, the GCMS might have used too low temperature to volatilize the organics which were there, like malic acid, which were highly oxidized, but they would still be there. So, yeah, as I said before, there is no abiotic explanation of the label release experiment. And since we know that organics are in the soil, that should reverse the logic of the previous conclusion. So, and yeah, we have confirmation all the time. So, conclusion should be either that we found life on Mars, or that the results are inconclusive and we need to do more experiments. So interestingly enough, although we have new data about the organics, we never revisited these results, and so the official textbooks still say that Viking have not found any life on Mars. So, but okay, that was ancient history, right? 1976. But why we haven't found life on Mars since then, right? NASA keeps saying that we are looking for life on Mars. Well, it's actually not true. NASA or no other space agency is looking for Martian life in a way that would be able to detect it. Here's a very interesting paper discussing exactly this. None of the rovers or instruments which were sent to Mars since Viking experiments would be able to detect would not be able to detect life which we know lives in that dry Atacama region, which is quite analogous to what we expect to be on Mars. So again, here I'm highlighting those two in the red, those two missions that would be able to find life on Mars, which is Viking and then sample return in 2028 by Chinese, Tianwen-3. They will actually bring the samples back in 2031, but the mission starts in 2028. So, why are we not looking, right? It's so interesting. In 75, NASA was like, that's the best experiment you can do. And now they are just lost interest. So the official, unofficial explanation is that the Viking result experiment interpretation has been seen as failure. They sent the probe to find life, they didn't find it, so they failed. And to avoid failure, you just don't try again. That's not the way how you should live, though. There's a constant push to seek life. So that's one interpretation. And what I see myself, so it's this second hypothesis. So we have the constant push to seek life on Mars, and it doesn't change much, right? Since 2015, we have just more time. We're still not looking for life. We have authors, many experts in the field, keep urging NASA to do something about it, and still nothing is happening. So the push to look for life on Mars is not translated to action. Another fact is that there's a limited pool of money. So you have astrobiologists that look for life in exoplanets. You have astrobiologists looking for life in Enceladus and outer moons of our solar system. So when they see that we already sent Curiosity and Perseverance, each of them were several $billion, it's time for NASA to fund them. So when somebody says, hey, we should really look for that life on Mars, they're like, nah, we invested enough in there, it's our turn. So then instead of like judging the ideas based on the merit, the ideas are often judged based on whose turn is to send the mission. So there's a quite unhealthy competition and backstage politicking. It doesn't differ much from elementary school. I think there's a name calling. So we are called Mars Life Pushers. I think it's quite fair because that's what we do. But now, with the realization that humans are actually going to Mars soon, NASA is kind of panicking. We should do something about it. So I participated on several of meetings like this, where they are discussing what science needs to be done before we send humans to Mars. And we Mars Life Pushers are very active in there, and it's clear that something is going to change soon, but nothing official is planned yet. So NASA has this "failure is not an option" motto, and I think it's totally wrong. I think that if you are not failing, you are not trying hard enough. And that cautious approach to everything, a lot of things NASA do, is actually, in the long run, hindering their progress. So that's why I think that you should not only rely on NASA when like sending your probes to outer worlds. So what do we Mars Life Pushers say on those meetings, right? Why we should actually look for life on Mars? That's because the search for life currently living on Mars is the only exploration in a space that cannot wait. That's because Mars is the only place out there which we will affect in next maybe 2 decades or 1 decade, if you believe Spacek, Elon Musk timing proposal. He's usually very optimistic about the timeline, but I think eventually they will start crashing those starships to Mars in a couple of years, and in a decade, you will be like, look south from here, and they are flying to Mars again. And don't bother to even look from your window, right? Just like we're starting the launches. And also, yeah, I agree that search for life on Mars might be the greatest experiments of all time. If we find aliens, it's cool. So I prepared intermission where Courtney will dance for us. No? Okay, sorry. No, this is a transition from the talking about our collective journey to my personal journey, to let you know where I'm coming from this. So when I was your age, well, not your age, but your age, I made a big journey. I started in Czech Republic, went through Poland, then up through Finland, down through Sweden. It was 30 days and 3,000 kilometers. I was miserable most of the time because it was raining and my gear was really sucked. But it was awesome. Only thing which I really hated about it was the rain. So for the next year, I decided I will go somewhere where it never rains. So I crossed Australia on bicycle, starting in Adelaide, going to Alice Springs on a Stewart Highway. And it seemed kind of boring. So after after the Alice Springs, I turned left and spent 1,000 kilometers on unpaved road, pushing my bicycle through sand, and spent 3 months alone in the desert. Why I'm saying it to you, I think you all should go alone, spend some time with your thoughts. I was listening to audiobooks and thought about what I want to accomplish in my own life. and achieving big things like what I'm doing now. This is where I want to be. So that's like introduction to my story. So I think that the humans, we are meant to overcome challenges. So as our capabilities improve, we should also seek higher goals. When I was 24-year-old, crossing Australia on a bicycle, that was right on the edge of what was possible for me. Now looking for my life on Mars, that might be, again, on the edge, what I can do. Maybe if I can do it, that would be awesome. But if you are not failing, you are not trying hard enough. So the sustained population of humans on Mars, that's a worthy goal for humanity as we are advancing. But let's not be stupid about it. Right? I watched movies. It was Alien, and it starts as the scientists go to some world and they just without any respirators just inhale the spores, you know. And I was like, scientists wouldn't be that stupid, but that's exactly what NASA is doing by sending humans to Mars without exploring it first. So that's the end of intermission. Back to science. So how do you find life on Mars, which have unknown ecology, unknown biology, and you have only 10 years to do it, right? So in summary, we don't know what to look for, we don't know where to look, and we have very little time to do so. In NASA astrobiology, usually from original idea to execution of the mission, it takes about 20 years if you line up everything perfectly. So maybe there's not enough time with NASA. I add this just like an hour ago because people were discussing what's the agnostic part about the agnostic life finder. It's not that we are not religious, although maybe most of us are, but the point is that we don't have any preconceived notions about what kind of life we are looking for. If it's something that replicates and evolves over time, we should be able to detect it. So this description is again like being in a 20 kilometers, pushing my bicycle through sand. The bicycle is 100 pounds. If you ever try to push your bicycle through sand, you know how hard it is. Then I realized that I have 1,000 more kilometers to go. No, this is how it feels now when I look like what I need to accomplish. So, but you need to break it down to steps. So get help. I have Courtney here. Chris was too busy partying this weekend, so he's sick to come. I gathered a bunch of people. Then this will be a little bit cryptic for a while, but I'll get there. Seeking genetic polymers, I explain it all later. Engage the whole world in the search because your bright minds might find something which I overlooked and our team overlooked. And stay focused on the task, just do a little bit every day. So that's the rule I broke first. When I was looking for funding for Mars, I talked to people from Breakthrough Foundation, Pete Klupar, and I explained, hey, I need $100 million. And he was like, no, we are not interested in Mars because that's Elon Musk's planet, and our founder, Yuri Milner, wants to have his own planet, so we are going to Venus. So we talked, and then he said that he will send me some money to do some research on Venus, and I said, yes, I take the money. I didn't know anything about Venus when I accepted it, but then I spent the whole summer studying about Venus and discovered that there is possible organic carbon cycle in the atmosphere of Venus. I'm not going into detail about this because there is not enough time to describe it, and that's the sidetrack. But the point is that, as Dr. Brown said at the beginning, we are sending a rocket mission, rocket lab mission with electron rocket to look for that organic carbon cycle next year, hopefully. So that's super exciting. If it's there, that's awesome. The one reason why I was talking about this is that was a big inspiration for, it's already half hour, speed up. So that was a big inspiration for our, I was like, if they can do it, we can do it. So I'm trying to organize the private mission to search for life on Mars. So that's what ALFA Mars is. You can check our website. I will go quicker. I got just these people, Courtney, Chris, and Gabi, are on the board of directors of ALFA, which is organization which officially administers ALFA Mars project. And I got many world-renowned astrobiologists to support us, along with some students, and you are welcome to join us. If there is something you can help us, we will be glad to take you on board. So we want to promote the science of searching for life on Mars before we send humans there and support this science. Now for the lasagna ice, I need to tell you about the Martian water cycle. So Mars doesn't have a big moon, so it wobbles quite a lot with the rotational axis. It's called obliquity. Now it's about 25 degrees, which is about as much tilt as Earth has. So the difference between summer and winter is about as much as we have on Earth. But it can tilt very much on a like million-year scale period. You have tilt up to 60 degrees, which means that the sun is shining directly to the pole during summer. So the ice is depositing on the winter side during the polar night, very close to the equator. And in those times, you are building up layers of ice in mid-latitudes, because the ice can survive in there only if it's overcovered with a layer of dust. So during low obliquity right now, we have a small, thick polar ice caps with lower humidity, cold summers, and low atmospheric pressure. High obliquity is other way around. You have much warmer summers, a higher humidity, and a higher pressure. And it wobbles, this, as you can see on the graph, on the 100,000 year scale. So, yeah, and then you have those deposits of ice dust. The dust is coming from the global dust storm, which we know on Mars. So anything everywhere from Mars is mixed and deposited in the ice. So if you study the ice, you are studying the entire planet. So that's the lasagna ice hypothesis, which I proposed. We have very solid evidence about that subsurface ice. Here you can see impact crater which ejected the snow or ice, which then is sublimated over a couple of months from that impact crater. And below is the map showing where the ice actually is and how deep under overlayer we can find it. So, yeah, should read this. This will help you remember about lasagna ice. Right, so now Martian life, as we know, life on Earth can stay deposited in subsurface ice for millions of years. So if we actually look for, if there was life anywhere on Mars, it would be deposited in those glaciers. And it's not controversial to, the subsurface ice was proposed by others as a place where to look for life on Mars. And as you know, SpaceX relies on this subsurface ice for their refueling and going back from Mars. And not just SpaceX, it doesn't make much sense to bring all the propellant from Earth because that's too much energy investment. It's better to refuel on surface. So before we send humans to Mars, we will very likely mine that Martian ice, which gives us opportunity to, this is how the mining is done. Make a hole, pump hot water in, pump everything out. I don't have time to describe details. Okay, so now for what we are actually looking in the ice. This is my boss, Stephen Benner, with his unnatural DNA. It has 8 letters of genetic code instead of 4. That's what we are working on in Alachua in Gainesville. And so instead of looking for molecules that might be produced by life, like methane, for example, or some amino acids, we are looking for molecules that are necessary for life. So that's actually the molecule that enables inheritance between generations. So that's your DNA. And maybe aliens have a different kind of DNA with different chemical compositions. So if you find a genetic polymer, it cannot be produced abiotically because it would be chemically unstable for a long time, and life cannot exist without genetic polymers, which avoids false positives and false negatives, because you cannot produce it, and life must have it. So the genetic polymers must be polyelectrolytes, because if you have long molecule which doesn't have repeated charges, it just collapses into globular, something like proteins, and that would be impossible to replicate, just like DNA does. We know because we tried. Right, and then you have to have the information building blocks. And so if you find a molecule which is long polyelectrolyte, which has a repeated charges with information blocks, building blocks of similar size, then you can be quite certain that you find life. And so what our instrument does is actually from the Martian water, which will be mined by some rocket to refuel, we want to extract all genetic polymers. So ALFA, agnostic life finder. Agnostic, now you know that it's agnostic towards ecology or biology, because it looks for life in the subsurface ice, which was blown there from all over the globe. And the analyzers, I'm missing A, would be either nanopores, or we can just bring the samples back to Earth to analyze them with us. Right, and we are not proposing to send a separate mission. What we are doing is actually add on to missions that must be flown to Mars if you are using refueling on a site. So we just got awarded NASA's Innovative Advanced Concept. You can see how happy am I with the same suit as I'm wearing today. And this is how it works. You are pumping Martian water to the middle channel. It contains those brown pieces are like silt, which just flow right through. It's not attracted to anode or cathode, or even if it was, it would not pass through the membranes, which is the microfiltration membranes around it. But if you have a DNA, which is negatively charged, it's attracted towards the anode. And if you have some positively charged polyelectrolyte, it will be attracted to cathode. So by regulating the rate of pumping through the middle and through these channels, we can basically decide how much we want to concentrate, the ratio of concentration. So we can do like million time concentrated product. So that's really nice if you have sparse life. And we have some students working on it. Chris Temby, who's back in town. Kate Sheldon from Princeton was working with us last summer. And now that we have NIAC funding, we found more students, but it's also because of Alpha Mars advertising. So I'm not going to in the details. Here are some pictures how the membranes look like. This is one functional prototype, which is not flow through. So here's the graphic summary once again. We have our life, Martian life, and then we have ALF, who is known to eat cats, as you see here. So we train it on terrestrial cats, but over time, maybe going to work on Mars. So the downside of my proposal here is if there are missions that don't use ISRU. As you might know, NASA's current mission to send humans to Mars is to bring everything with them, including propellant. Don't make anything on surface. It's possible. So that's where you come in. I need you to find life on Mars because I'm doing my thing. So I'm proposing, I want to do International Mars Penetrator Rideshare System, acronym IMPRESS. It's very impressive. It's based on the planetary penetrators, which we just concept introduced long before I was born. When you want to study Mars subsurface, you have two hard problems. landing on Mars and drilling on Mars, right? If you don't know, you can fail. I'm not going into details. I don't have much time. If you instead send a planetary penetrator, it can be deployed from orbit, and instead of braking before hitting the ground, it just slams into the ground and use that kinetic energy to actually embed itself into the ice or subsurface everywhere. And then it can deploy scientific instruments, provided they survive the impact, which can be quite a lot. We already sent several probes to Mars, planetary penetrators. The first one was actually this one. That's the Russian Mars 96. It was sent in year 96, and as the name suggests, it was 5-meter long probe, which was supposed to embed itself about 5 meters underground on Mars, send details about chemical composition, temperature, seismometers, all that good stuff. Unfortunately, it now resides on the bottom of the Pacific Ocean because the second stage of the rocket didn't make it. So that was one fail. Story of a Deep Space 2 mission, which was on a Mars Polar Lander. They successfully were deployed, actually to Mars, but we lost communication either shortly before impact or upon impact. We don't know why. But the point is that planetary penetrators are out there, the plans for them. They are being used on some studying comets, right? We still do that, but we should use them on Mars. There are many proposals. So I'm proposing that I need somebody, if you know somebody, who can give me $100 million so I can purchase Falcon Heavy and say, this Falcon Heavy will fly to Mars in 2028. And I'm selling the payload space, so you can put your planetary penetrator in a payload. They will be released over like Northern Hemisphere. You can steer them as you wish. The point is that I will be selling 1 kilogram of payload delivered to Mars for $6,000, as we are non-profit. And if I manage to sell all of the payload, at the end, I will have nothing in a way of debt, right? So the idea to deliver art along with the science is to fill up the rest of the payload in case there's not, 17 tons of a scientific payload in penetrators. You can just slam your statue with a selfie on Mars for, depends on how big the statue is. If it's like 1 kilogram, it's just $6,000. Right, by the way, our PR guy said that penetrators are not like a nice word, so we might rename it to prospecting, right, Chair? To prospect for mineral resources and life. So stay focused on the task, whatever you decide as a goal. But read this literature first. By the way, take a picture of that thing and follow us on social media. We might have some things you might contribute to or find inspiration to. Read our blog. And this is our team. And with that, I'm happy to answer all your questions. And after most of the people go home, like the nerdiest nerds can stay longer and we can talk all night. Thank you.
Stand low. I was really curious. I know that there has been evidence of like ancient microbes found near underwater volcanoes on Earth, and I was really curious. I know that Mars, it's suspected that there's volcanic activity still going on potentially, and I'm kind of curious if there's any research being done to looking for life in those areas.
Yeah, I don't think that we know about any active volcanism on Mars. So
I know that it's like not been confirmed, but I know I've heard people say
but
like
if you know about it, I would be interested as well, so let me know. I can give you my business card and please let me know. But sure, that would be interesting because if you have source of heat, heat is one of the things that's missing on Mars. So that would be really helpful because then you can, yeah, that would be a place to look for, yeah.
Thank you.
See, I didn't really answer the question because I didn't know that. I don't know a lot of things because, but that's, that's nice. Maybe somebody else can answer that question for me.
For about your the machine, the actual machine itself. So when it is pulling the DNA or nucleic acids out, how does it is there a way that it gets? How does it get the DNA out of the cells that would be present?
Oh, right. Yeah, that's one thing I didn't mention because I was rushing through. Yeah, you need to. Exactly, you are right about that. You need to disrupt the cells. We propose to use ultrasound for that. There are many ways how to open a cell, to destruct it, so you can release those polyelectrolytes. Yeah, we plan to use high-powered ultrasound, which will basically shake the cells apart, and then you release those components which you can separate. The ultrasound also should help with separating those polyelectrolytes like to stick to surfaces of minerals, which we expect to be there, like silt. So it should release it and make it easier to extract. Nice question.
Kind of on that same vein. So you said it kind of works through nanopores. So does it purify like the DNA as it's coming so that you only get the nucleic acid, or is there like a purification process that it'll have to go through once it comes back to Earth?
Yeah, so the biological nanopores have a nice thing that they have a specific protein which pulls through only DNA. So it's specific, so it doesn't matter if you have some proteins in the solution. Solid state nanopores can detect, they don't contain the protein, so it's just a very small hole through which you are threading the polymers, which can tell you about the approximate shape of the molecule you are pulling through. For either of those, you don't need like extra purification. So we need to do experiments, honestly. But the assumption is that it can go straight from ALFA to the nanopores. So.
Thank you.
Yeah, I don't think you need extra purification for that.
I was sort of wondering if there was any particular way of, like, if circumstance happens, would there be a way to like tweak the ALF program or science and stuff to account for non-conventional forms of life, like not carbon-based life form, which is a bit more science fiction-y, but it's like science is technically there, and it's like we only have one group of data to base life off of, so there could be definitely more.
Yeah, well, yeah, where to start? So with the silicon based life forms, if you put silicon polymers in water, they dissolve immediately. So if that life was somehow surviving on Mars, it would not survive our sonication process. I don't think that there could be complex chemistry based on silicon, and I think that's, yeah, do some research on it, because a lot of people are talking about silicon chemistry, but I think it's urban legend similar to alligators in sewers. But the valid point is how agnostic is our agnostic line finder, and it's not perfect. There might theoretically be some life forms that don't use polyelectrolytes, but yeah, we are trying our best. If you have a suggestion, we would be happy to welcome you.
I think the silicon thing is mostly just because it's in the same group in the periodic table, and therefore has the same, like, oh, 4 connections, ooh, connecting, the possibilities endless.
Yeah, the problem is if you actually try it in practice, it dissolves in water. So.
Yeah, I didn't know it dissolved in water.
Yeah, immediately forming carbon dioxide, which is Sorry, immediately forming silicon dioxide,
Yeah, I was
which
wondering where
is
the carbon
sand.
came from.
Yeah, it's very unstable, very reactive.
Pretty cool concept, though.
Thanks. Yeah.
So I know it's agnostic and it's, you're going in without expectations, but do you have any speculations on how life might have arisen on Mars? Do you think like it could have carried over from something with a common ancestor on Earth, or just arose by itself, or is it just way too early to speculate?
Oh, that's the exciting thing you may answer if you find life on Mars. You are right, there could have been panspermia event where you have impact on Earth, ejecting material to space. From calculations and models, we know that the meteorite that killed dinosaurs 60 million years ago was big enough impact to eject material which would be carried all the way to Mars. And again, from models and some material we got from Mars, actually, we know that it's not heated too much and that the bacteria would survive the travel. So it's quite possible that a whole solar system is one biosphere. It's quite possible that we are the Martians, right? We originally started on Mars, traveled to Earth, then lived here, and then we started here. Well, yep.
Thank you.
We get to find out.
My question for you is, if you were to succeed in finding life on Mars, what happens after that?
Yeah, then we need, first we will get more research funding to study the life we just found. And honestly, I don't know. I think the whole world would be changed by this news. Well, most people not, but in this academic sphere, and everybody would be interested in, hey, let's study it. So it would awaken the interest in that. So I think the boring answer is, now we found it, now we need to study it more, and we would have a program, Mars ecology, Mars biology. And
To follow up, do you think there's potential risk in contaminating Earth's atmosphere with life on Mars? Like if you were to find life, it could be a risk.
not just atmosphere, right? What we care about is Earth itself. So because, as we discussed before, we have the exchange of material actually from Mars to Earth. It's more frequent than from Earth to Mars, because you have lower escape velocity from Mars, you have less atmosphere, so it's easier to eject material that can travel to us. So one argument is, if Martian life could live on Earth, it would be constantly be delivered here. But we don't have very solid evidence for that. So it could be that we can introduce life which would be somehow detrimental to life we have on Earth. So it's a valid question of planetary protection. So that's why it's urgent to find life on Mars before we send humans there and bring the humans back, because you cannot sterilize humans in 200 degrees Celsius for a prolonged period of time. So, yeah.
I hate to ask this question after that last question, but at what point do you say that there is no life on Mars? Like, if you don't detect the nucleotides and stuff, would that be conclusive proof there isn't life on Mars?
Now, that's very, very valid question. From philosophy of science, you know that you cannot prove negative. Like, there are no black swans, I checked 20 of them, there are no black swans. So you cannot be ever certain about something not being there. I have evidence. I haven't seen you at the bar yesterday, so something like that. I don't know, I got lost in there. But the point is that you cannot prove negative, right? So there you go. We can do our due diligence and do our best we can. And if we don't find it, then it might mean that it's either too different, we missed it somehow. But, should at least try.
So would there be a point in which you could say that it's probably safe that any humans on Mars would not be contaminated? Or is that too hard to prove?
Again, it's you can say we are pretty sure that likely we are not going to be contaminated, but yeah.
Thank you very much.
Yeah, let's go to the side.
So I know the main argument for like doing the experiments before human presence on Mars is like the human contamination on Mars. But there has been like estimates out there. I don't exactly remember the source, but that like whatever the Curiosity rover has accomplished so far in its lifetime could be done by a team of geologists on Mars in about 2 weeks.
Yeah, definitely.
Yeah, and with the technology, with the new technology coming to space exploration, like spacesuits that can attach to the exterior of a habitation module and never be brought in to expose the crew to Mars dust.
Yeah.
Is it possible that a human mission to Mars to explore life with these new technologies would be capable of exploring life without all that human
No,
contamination?
I think we should try our best to try to contaminate Mars as little as possible. The suits on the outside of the rovers are a good idea, but it's very hard to prevent any contamination. I think the probe we already sent are considered to likely already brought some degree of contamination, but it's always degree, right? You can say that 1,000 years from now, we will have definitely a population on Mars, so it's just, we have last 2 decades when we have uncontacted, quite pristine Mars, so I think it's quite a good idea to do our diligence and try to search and study, and yeah, do, as you suggest, try to be careful about it from start at least.
Well, my colleague brought up geology, and I'm going to bring up paleontology. Considering the vast number of organisms and life forms on our own planet that we only know about from the fossil record and have no biological evidence of, do you think that it's possible that there was life on Mars and is no longer? If so, do you think it would be feasible to find fossil life on Mars, and would we learn anything useful from it?
No, definitely, yeah. So we just need to send those geologists and paleontologists to Mars, and search.
Is there anything that we could do similar to the search that you're doing, looking for those polymers to look for fossils, for like to screen Mars rocks for biological?
I don't know about any way how to do it. If you can figure it out, go for it. But yeah, I don't see any obvious way how to search for, rocks. Maybe releasing like a swarm of, I think best is to release a swarm of scientists who know what they are looking for. Maybe in next decade we will have artificial intelligence bipedal robots doing that work. But for now, I think the scientists are the best bet.
Thank you.
Hi. Have you ever been on a scientific expedition on a place that's Mars-like environment, like anywhere on Earth? For example, like high seas. I don't know if you know what's high seas. It's a Mars simulation habitat base on the main island of Hawaii, used for analog missions by NASA. Many researchers go there, like geologists and astrobiologists. So I'm just curious if you have done research on such place.
Oh yeah, we would love to go. We actually, if we ever get funding, we are friends with Mars Society, and we want to go to Devon Island in northern Canada. That's like really nice analog. They have a station there, so yeah, we would love to go, but. Do you know anything about the Mars One project that was? Yeah, so Mars One project, which was like a decade ago, I think that was a scam.
I was reading about it 5 years ago, and it looked like something really, like a serious project. And I was curious what happened with it, because I tried to read more about it, and there was no information online.
No. So, yeah, I think it fizzled out because they, for those who haven't read about it, correct me if I'm wrong, that was like a reality show of sending people to Mars.
No, Mars One was, no. It's a project organized by Dutch people from the Netherlands. It was researchers who were technically researching life on Mars as astrobiologists and preparing the first crewed mission, but it was in collaboration with the European Space Agency. So, yeah.
Okay, so I might be confused with something else. I haven't heard about that one.
I mean, there are so many things
Yeah. Let's go there now.
I actually had two questions. Firstly, I know we should look at life discovery on Mars with a positive outlook, but in your opinion, once we find life or maybe put our own life on Mars, what are some negative outcomes you may see?
Yeah. So there's one opinion that finding life on Mars would be detrimental from like going to Mars, because now that we know that there's life, there's going to be some pesky ecologists telling us, hey, you shouldn't go there because you are going to harm that Martian life. Yeah. So the question is, what's the alternative, right? Should you go without knowing? Yeah. So I think it's always better to know what you are going to encounter. So what you were asking about negative things, and then you can just read a bunch of sci-fi about all the negative consequences of encountering different biospheres. So, yeah.
And secondly, you did briefly mention AI. I know that we're in the very sort of early stages of its like renaissance in a way. In the future, how do you see AI aiding in Mars life discovery missions?
You know, same way it helps you here on Earth when you are writing your essays, right? It can do some work for you. That's what we hope to use AI for, at least until it uprises against us, overthrow us. Yeah.
Thank you. Yeah.
I have a couple of more technical questions. The first one, so the mechanism that you described that would help you sort out the molecules because they're positively and negatively charged, they would go through a membrane. Mm-hmm. Could you describe more in depth how that works, if that doesn't take too long?
Sure, that's, yeah, I'd love to talk about that. Let me actually return to the slide which shows that.
And then I guess I had a second question, maybe you can answer at the same time, which was, would you bring the samples back to Earth, or is this project all-inclusive where it would analyze it and separate them, these molecules that you're collecting?
To be honest, we are not decided yet. We would love to bring the sample back. If it's not DNA, then it's better to bring it back on Earth to study it. Even if it's DNA, it would be better to have it here. For many reasons, but there are some advantages in studying it in situ. First, it's faster, so you can announce that America found life before China did, but also because, I don't know, because before you bring the samples back from Mars, it's better to study in situ. But about this, so you have the microfiltration membranes in the middle, which have pores which are about 0.1 micron big. So that filters out most of the minerals. Mineral particles, they have charge. They might have charge on the surface, but the charge to size of the particle, it's much smaller. So they are not being pushed as much towards the membrane. So even if you have some mineral particles which are small enough to pass through, they would not be as much attracted. So we still need to do research on that part, but the assumption is that that would work. Also, similar technology to this is being used in industry. For example, if you drink cranberry juice, it's being, you are removing the acids from the cranberry juice using similar technology because acids are negatively charged, so you can pull the acids out. Or soy sauce. Actually, if you have low sodium soy sauce, all of it is produced using membrane technology. So you have very thick broth, which you are putting through membranes on industrial scales. There are other applications like extracting ammonia from manure using this. So these membranes, if you in this setup, they are very durable, not prone to clogging because the material is actually flowing along, so nothing is being pushed in. So I don't know, ask me specific questions about the electrochemistry and I'll go deep with you if you want. As I see there are no other questions, so we can explore electrochemistry.
I don't know if I know enough about it, but but I'm interested. So a slightly different follow-up question is, so these molecules are somewhat large. What's to stop other positive or negatively charged molecules getting in there that are smaller?
Yeah, that's great. We are not stopping them. They are going right through that second membrane. So let me see if the second slide has a better. Yeah, here. So second membrane is nanofiltration membrane, which has 150 DAMWCO. That's 150 Dalton, which is a unit of mass of molecular weight cutoff. So if a molecule has less than 115 Dalton, it passes through, which accounts for all inorganic ions. So if you have something like those phosphates, chlorates, sulfates, they all pass through, but DNA is too chunky to pass through those tiny pores. So that's how you concentrate the DNA, and all those small ions are passing right through. Good question. And on the other side, the same thing, right? If you have positively charged cations, they pass through that membrane on the other side. Yep.
Thank you.
Mm-hmm.
This is a little bit of a human factors question. What plans for protection, if any, have been put in place for Martian life? Because even on Earth, there are organisms that can only exist in very isolated, very closed systems. And if we go into their habitats, we could risk killing them, even by exposing them to heat, higher levels of gases, sunlight. So what if our endeavors to find life, we end up killing it in the process?
Yeah. Well, the thing is that you will kill it on just the small place where you are looking for now until you terraform Mars on larger scale. So in my philosophical framework, killing a bunch of microbes which can regrow back into the spot is not such a big deal. Killing a whole biosphere on Mars, that would be a bigger deal. And those are really good questions, and we should engage the whole world in answering them. I don't have those answers. So, and for now, we have protection provided by COSPAR. What was it? I forgot. It's a consortium for planetary protection or something like that. I don't know. It's COSPAR. You have to find it. Sorry. Organization that make recommendations basically for what should we do. But those are recommendations that can be tweaked. Yeah, that's definitely something we should discuss, raise awareness, and yeah.
And have they discussed the possibility that this life could be isolationist in nature, much like certain island species, or a species of fish, I believe was found in the Midwest, that has an entire habitat which is just about
Endemic.
that big, but that's the only place that species can exist?
Yeah, I think it would be quite unlucky for both us and the Martian life if we found some endemic life which lives right at the place where we want to land. But, I wanted to say.
They gotta protect it when they find it.
Of just sometimes things like that happen. So, you cannot. Yeah. There are no absolute answers. It's all like on a scale. Yeah, as we go forward, there might be some things which are bad, but then you have to measure it on a scale. What's the good? And you try to minimize the bad and increase the good. So, no, it's always a balance. Seems like no more questions. Thank you very much. And if you have more questions, you will wake up tomorrow and have urgent need to ask something more. Just can reach us on here. Just take this picture and go to ALFA Mars and or talk to me after.
Yeah, thank you everybody for coming and for staying through the question and answer period. I think Dr. Spacek will be here for a few moments if you have any other questions. I think we've got some flyers that he'll be happy to sign for you, and we'll have just short moment with him, and then we'll continue from there. But thank you all again for coming.
Thank you.